Hello, this video tutorial is about the scientific method, so let's think it like a scientist. Well, in order to start, this scientific method is divided into several steps. The first of one, that is the observation. An observation is something that really calls your attention when you were looking at a phenomenon. In this example, for example, I have that you are looking some salamanders near a pond and you notice that they have curved and not straight tails. So this event just calls your attention. This is an observation. It's quite different from being just looking at something. When you are making an observation, you are getting into a detail that is really particular and that generates some questions in you. Then, after you have done the observation, you jump to the second step of the method, that is the hypothesis. A hypothesis is a suggested solution to the problem, or is an educated guess. In order to have a hypothesis, this hypothesis must be testable. Otherwise, you cannot run out the experiments and then you will be out of the scientific method. In order to make the hypothesis, you can make statements that are usually written as if or then. Okay? And in the hypothesis, you are predicting an outcome. This outcome is the one that is going to be tested. In the example of the salamanders, then, your observation was that some salamanders have curved tails. Okay? The hypothesis could be that the salamanders have curved tails due to a pollutant in the moist soil where they live. So in this case, you must or you are able to test if the pollutant actually is having an effect on the shape of the tails of the salamanders. Then you have a hypothesis. What's coming next? The experiments. So an experiment is a procedure to test the hypothesis. It's the most exciting part of the scientific method as well. In the experiments, you must take into account that the experiment has variables. What is a variable? It's a factor in the experiment that is being tested. The experiment is going to be valid or good only if you are testing one variable at a time. This is in order to have comparable data because you are going to run the experiments over and over again. You are going to have several trials in order to reach validity in the testing that you are making out. So please remember, just one variable at a time. Well, when you're constructing your experiments, you have controls and variables. We are going to take a look in more deep. Well, these are the rules in order to follow an experiment. So the experimenter may changes one factor, okay? And by changing one factor or one variable, he is going to observe or measure what happens in the other variable. This is how it works. So you are in control of one variable in order to see the effect that is being produced over another thing that you are looking at or that you can measure. Let's talk about the control variable. So the control variable is the variable that the experimenter, okay, is going to manipulate. So the experimenter makes a special effort to keep other factors constant so that they will not affect the outcome. It means, for example, that if you are going to observe the effect of temperature on the conditions of an ecosystem, the control variable that is the one that you can change is going to be the temperature. So it's the one that you are going to manipulate. That's the control or independent variable. Okay? So the factors that you are going to control are control variables. What is the purpose of having a control? Well, the controls are used for comparison and they are not being tested. Okay, so you are not going to affect that variable. Actually, this is the one that is going to make you compare in order to have the final drawing conclusions. There are other variables. So we have the dependent variable. The dependent variable is the factor that is going to be measured or that is observed while you are changing the independent variable. We have an example of the effect of temperature on the gender of baby alligators. So I can change the temperature, that is the independent variable, is the one that I'm changing. And what I am going to observe is the effect of this change in the temperature on the gender of the baby alligators. So maybe if it's cold, it's going to be produce female, and it is hot, it's going to produce male babies. So the gender in this case is going to be the dependent variable. That is the one that is under the control of the temperature, that in this case is the independent variable. Well, let's see some examples about controls and variables. Then suppose you want to figure out the fastest route to walk home from school. Then you will try several different routes, and you are going to register the time on how long it takes, okay, for you to go home by each one. Now that you are interested in finding the route that is fastest for you, you will do the walking yourself. Another way is to have other people making the walks, but in this case we are going to work just with you. Then which are the variables that you have in this experiment? So varying the route is the independent variable, is the one that you are able to change. You are the one that is looking at the different kind of routes, so you are in control of this variable. That's why it's the independent one, because you are changing it on purpose. Now, the time it takes is the dependent variable. Okay, so you are changing the route, and by changing the route, you are affecting the time that you spend by walking home. Okay, then the time that you are recording, it's going to be the dependent variable. What is your control? Your control is that you are going to be the same walker no matter the route you're taking. Okay? Okay, one more thing. As I told you before, it is best to make several trials with each independent variable. Why? In order to reach validity. If you have only one register, you can be skewed or you can be biased, and you may reach a wrong conclusion about your experiment. That's why it's quite important to make several trials in order to consolidate the data. Okay, this is about valid experiments. So remember, in order to have a valid experiment, you need two groups, okay? The control group versus the experimental group. And you are testing only one variable at a time. Please remember that is quite important. After you have run your experiment, then you can go to the next part of the scientific method, that is the data. The data are the results of the experiment. They could be of two categories: quantitative, that is rather in numbers, or qualitative. The data must be organized. How can you do in order to get it organized? You can register your data into charts, tables, graphs, okay, in order to have them and to look at them in an orderly way. After you have collected all your data and you have it organized, you can start with the other step, that is drawing up conclusions. So the conclusion is an answer to the hypothesis that you were posting in the first step, and is based on the data you obtained from the experiment. So based on the data, You may accept your hypothesis or you can reject your hypothesis and you retest. Okay? If the data doesn't support your first hypothesis, then you retest or you change the hypothesis for another one, and you once again are going to fulfill the complete steps. The retest, as I was telling you, is in order to verify the results. Every experiment to be valid has to be retestable. It means that you must be able to make several repeats and have or obtain the similar or same results. And also, if another person wants to make once again your experiment, it also has to be repeatable. Okay, that's the richest part. Let's take a review. You are going to solve a problem. Then you first need to identify a problem. To identify a problem, just state the observations about the problem. Remember the things that take out your attention. After that, you form the hypothesis about the problem by making statements going with if and then, and please be sure that this hypothesis must be testable. In the fourth step, you are going to design an experiment to test the hypothesis. Keep in mind that you need a control group and an experimental group, and that you are going to test only one variable at a time. After you have designed and running the experiment, you collect the data in an orderly way, you process the data to reach and form a conclusion. If the conclusion supports your hypothesis, you can move on onto another new observation or a new hypothesis. But if the data is not supporting your first hypothesis, you need to retest it, okay? And you need to make a new hypothesis that better fits with the data you obtain. Okay, this is the end of this presentation. Hopefully you have enjoyed it and you know a little more about this